Today we will talk about the mechanism of DNA replication. We have already studied what the replication is. It is simply a DNA dependent DNA synthesis, simply the synthesis of DNA from the DNA molecules. It is called as DNA replication. And this DNA replication, it is semi-conservative. That means DNA molecule has two strands. Out of two strands, one strand serves as a template for the synthesis of new strand. That means in semi-conservative mode of DNA replication, DNA is half conserved. Regarding the today we will discuss mechanism of DNA replication. Regarding the mechanism of DNA replication, it is a multi-step process. It involves several steps. It is basically a multi-step process. It involves several steps. We have initiation of replication. We are going to discuss it in detail, mechanism of DNA replication. Initiation of DNA replication. Then formation of RNA primer. Formation of RNA primer. Third step is elongation of new DNA strand. Of new DNA strand. Elongation of new DNA strand. Then we have a termination of replication. Termination of replication. Then fourth, termination of replication. Then we have proofreading. Proofreading and cross-checking. It's called as fidelity of DNA replication. It is proofreading is responsible for fidelity of DNA replication. So we are going to discuss these steps one by one. First is initiation of mechanism of DNA replication. It is an elaborate and multi-step process. We have initiation of DNA replication. We have formation of Rna primer. We have elongation of this DNA strand. We have termination of replication, and then proofreading and editing, which increase the fidelity of the DNA replication. So first is initiation of DNA replication. We have initiation of DNA replication. Initiation of DNA replication. This DNA replication, it does not take place randomly on a DNA molecule, but there are certain specific sites, certain regions, what are called as replication origins, origin of replication. Origin of replication. These origin of replication, ORI, they serve as a site which will initiate the process of DNA replication. Actually, this ORI site, origin of replication, it is a particular sequence, a particular region on a DNA molecule which serves as a site for the initiation of the DNA replication. And this origin of replication, it is having a particular stretch of base pairs, that is adenine thymine. This ORI site is rich in adenine thymine rich stretch. This, I told you that DNA replication does not occur randomly on a DNA molecule, but it takes place at a specific region on a DNA molecule, and that specific region is known as origin of replication, what is called as ORI. This ORI region, this ORI site, it has a particular sequence, that is adenine thymine base pair. It's having adenine thymine base pair rich sequence. Now the question arises that why this adenine thymine rich stretch is present in the ORI site. Actually, this adenine thymine, it is having two hydrogen bonds. It is having two hydrogen bonds. Adenine having two hydrogen bonds with the thymine. Whereas guanine and cytosine have three hydrogen bonds. And it is easier to break the two hydrogen bonds because of the fact that for the DNA replication, the two strands of the DNA have to get separated, they have to get unwinded. And this requires the breaking of the hydrogen bond between the two strands. So it is less amount of energy is required to break the adenine thymine hydrogen bond as compared to that of the guanine cytosine hydrogen bond, because of the fact that there are two hydrogen bonds between adenine thymine and three hydrogen bonds between guanine and cytosine. So we can say that this origin of replication is rich in adenine thymine base pair sequence, and it is energically economical to have adenine thymine rich base sequence at the ORI site, because less amount of energy is required to break the hydrogen bond between the adenine thymine as compared to that of the guanine cytosine. So this is what is called as replicon. Replicon, it is simply the base sequence on a DNA, or we can say that it is a unit of replication. This replicon, it is the base sequence at the ORI site. This replicon starts from ORI site and proceeds up to the terminus site, and that is called as replicon. For example, we have a DNA molecule. This is a DNA molecule. If ORI site is here, this is ORI site, and obviously somewhere there would be terminus site. This terminus site, it represents where DNA replication will terminate. This ORI site, it is the origin of replication, and terminus site, it is the site which determines where the replication is going to stop. So we can say that the base sequences which start from ORI site and proceeds up to the terminus site represent the replicon. So replicon is a replication unit which starts from ORI site and ends up to the terminus site. And this terminus site represents what? It represents the site where the DNA replication stops or terminates. So it's called replicon. Remember that in case of prokaryotes, we have single origin of replication, while in case of eukaryotes, we have many origins of replication. So prokaryotes have single origin of replication, and eukaryotes have many origins of replication, because of the fact that this eukaryotic chromosome is greater in size, greater in length as compared to that of the prokaryotic chromosome. That is why we can say that to quickly complete the replication process in case of eukaryotes which have comparatively larger chromosome, this greater size of chromosome, there are many origins of replication in case of eukaryotes, whereas there is a single origin of replication in case of prokaryotes. Why this is because of the fact that eukaryotic chromosome is greater in size as compared to that of the prokaryotic chromosome. Which which is smaller as compared to that of the eukaryotes. So actually the size in case of DNA molecule in case of eukaryotes is greater as compared to that of the DNA which is found in prokaryotes. There is no chromosome in prokaryotes. Chromosome is basically a structure which is composed of proteins and this DNA, whereas in case of prokaryotes we have nucleoid or genophore, not a chromosome. So we can say that this single origin of replication in case of prokaryotes, and in case of E. coli, this origin of replication is called as ORIC. E. coli, ORIC, it is in case of Escherichia coli. It represents the origin of replication in case of Escherichia coli. And remember that in case of yeasts, origin of replications are known as autonomously replicating sequences. So what are these autonomously replicating sequences? These are basically they represent the origin of replication in case of yeasts, autonomously replicating sequences. So we have autonomously replicating, it is called as autonomously replicating sequences, what are called as autonomously replicating. So autonomously replicating unit, we can say autonomously replicating unit, autonomously replicating unit, autonomously replicating unit. In yeasts, the origin of replication are known as autonomously replicating units. In yeasts, the origin of replication is known as autonomously replicating unit. In case of prokaryotes, there is a protein, what is called as, in case of prokaryotes, a protein that is DNAA, also written as DNAA. This DNAA protein, it binds to origin of replication, and this when it binds to this DNA protein, when it binds to origin of replication in case of prokaryotes, it causes local unwinding. It causes, this is when it, for example, I will show it like this. For example, this is double-stranded DNA. When this is DNA protein, this DNA protein binds to the ORI site, it causes local unwinding or melting of the DNA duplex. So this DNA protein, it binds to the ORI site and causes melting or unwinding of the DNA duplex. It causes local unwinding, what is called as local melting or unwinding of DNA duplex. In case of if I will show you in case of this in case of prokaryotes, here I can show in case of prokaryotes what would be the origin of replication. For example, this is prokaryotic DNA, this is prokaryotic DNA, double-stranded DNA, this prokaryotic double-stranded DNA, prokaryotic double-stranded DNA. This represents prokaryotic double-stranded DNA. And for example, this is in case of prokaryotes. For example, in case of Escherichia coli, E. coli, this site, it single origin of replication, that is ORIC. This ORIC, it represents origin of replication in case of Escherichia coli, for example. And then this is what is double-stranded DNA. This is double-stranded DNA, and this DNA in case of prokaryotes, for example, in Escherichia coli, it is having a single origin of replication that is called as ORIC. And this DNA protein, it binds to, for example, consider that this is a DNA protein. It binds to ORIC site and causes the unwinding or melting of DNA duplex at this ORI site. And then in case of eukaryotes, we have many origins of replication. For example, this is an eukaryotic chromosome. Eukaryotic chromosome. This is eukaryotic chromosome, eukaryotic DNA. This is origin of replication, origin of replication, origin of replication. So in case of this is eukaryotic DNA, in case of eukaryotic DNA, we have many origin of replication, 1, 2, 3, because eukaryotic DNA is comparatively larger in size as compared to that of the prokaryotic DNA. This is also double-stranded DNA. So this DNA protein, it binds to the origin of replication and causes unwinding or melting of the localized unwinding and melting of DNA duplex at the ORI. Then another enzyme, what is called as DNA B. This is called as helicase. This helicase, it causes the separation of double-stranded DNA into the single-stranded DNA. That means if this is, for example, if this is double-stranded DNA, this helicase converts this DNA duplex into single-stranded DNA. That means it causes the unwinding or unzipping or separation of two strands of the DNA. This helicase. Helicase is the enzyme which separates the two strands of the DNA duplex. That means it would be like this. This is one strand, this is another strand. Actually, it's a DNA duplex, but helicase unwinds the two strands. Then there is another protein, what is called as DNAAC protein. DNAAC protein. This acts as an helicase loader. This DNAAC protein, it acts as an helicase loader. It loads the enzyme helicase on the DNA duplex, and then this helicase causes the unwinding of the DNA duplex, causes the unwinding of two strands of the DNA duplex. So this was regarding a DNA A protein, DNA B protein, and DNA C. Then there is when DNA would be like this. It would be like this. It would be like this because two strands are separated by means of enzyme helicase. This is one strand, this is another strand. Now these two strands, these single strands are stabilized by a protein that is called as single strand binding protein, SSB protein. That means now this single strand binding protein is called as single strand binding protein. Single strand binding protein. I will write here single strand binding protein. Single strand binding protein, SSB protein. These single strand binding protein, they will bind to the single strands of DNA, and their function of single strand binding protein is to prevent the re-annealing of this single stranded DNA into the DNA duplex. That means there is a great capacity for these single stranded DNA, the single strand of DNA, to get re-annealed, so that it will get again coiled and form the DNA duplex, but this single strand binding protein, it prevents their coiling and formation of DNA duplex, because it is necessary that we will just separate the two strands of the DNA that will act as a template, for the synthesis of daughter DNA molecules. Then as a result of this unwinding, there will be a type of a replication fork is formed. A replication fork. Replication form due to the unwinding of your unzipping of two strands of the DNA. A replication fork is formed. See, this is the replication fork. This represents what? It represents the replication fork. This is replication fork. This is DNA duplex, double stranded DNA. This is single strand binding protein attached to it. This is replication fork. That means due to the unwinding of the DNA, double stranded DNA, a Y-shaped structure is formed. This is just a Y-shaped structure. Y-shaped structure formed. This Y-shaped structure is known as replication fork, and this replication fork is formed because of the unwinding, because of the unzipping of the two strands of the DNA. Then because of the formation of this replication fork, due to the formation of replication fork, a coiling tension would be created here in the form of, there would be twists and there would be knots. When this replication fork will move ahead, as a result of this, there would be a sort of coiling tension, and what is called as supercoiling. Because of the supercoiling, what is called as supercoiling. A coiling tension is created ahead of the replication fork. This coiling tension would be in the form of what? Knots and twists. And this coiling tension is known as supercoiling. What is supercoiling? Supercoiling is when the replication fork moves ahead, it creates a sort of coiling tension. And this coiling tension is in the form of knots. That is in the form of knots and twists in DNA molecule. And this coiling tension is known as supercoiling. And as a result of this supercoiling, DNA replication, or we can say that separation of two DNA strands would not be possible. And then there is an enzyme that is called as Topo Isomerases. These enzymes, Topo Isomerases, their function is to relieve or to relax this coiling tension ahead of the replication fork. That means these Topo Isomerases, what they do, they either make the single stranded cuts in the DNA or in the double stranded cuts in the DNA. As a result of that, this coiling tension or supercoiling is relieved. And these Topo Isomerases use the energy of ATP hydrolysis for breaking the single strand DNA, for making the nicks, NICK, for making the nicks in the single stranded nicks or double stranded nicks, which will result in relieving of the coiling tension. So these Topo Isomerases, they make single strand nicks or double strand nicks. That means single strand nick means when only one strand will be cleaved, so this coiling tension is relieved. When double strand nick means when in both the strands breaks or being made by the Topo Isomerases, so that this coiling tension or supercoiling is relieved, and then this unwinding of DNA duplex can proceed smoothly. So this Topo Isomerase. And in case of prokaryotes, there is an enzyme that is known as gyrase. This enzyme gyrase takes the function of topoisomerases in case of prokaryotes. So topoisomerases are important for relieving the supercoiling coiling tension in case of double-stranded DNA during the replication process. Then we have got formation of RNA primer. Then formation of RNA primer. Then formation of RNA primer. Formation of RNA primer. This formation of RNA primer. Why this RNA primer is a short stretch of RNA? This RNA primer, it is a short RNA segment. This is a short RNA segment. Actually, this RNA primer is formed because of the fact that the DNA polymerase third, this is the main enzyme, DNA polymerase third. In case of prokaryotes, this DNA polymerase third, it cannot initiate the synthesis of DNA. So it means that this DNA polymerase, it can add deoxyribonucleotides only to the 3' end. Now, in order to make the environment suitable for the DNA polymerase, because DNA polymerase cannot initiate the DNA synthesis, so a short segment of RNA is formed on the DNA templates. It is formed at two sites, at the DNA template, at the free end on the DNA template, and at the replication fork on the DNA template, which will help the DNA polymerase to add the deoxyribonucleotides. It will be like this. We have, for example, I will explain it like this. We have this is replication fork. This is 5' end. This is 3' end. This is 3' end. This is 5' end. We know that DNA molecule is double-stranded. This is one strand. This is double-stranded DNA. Double-stranded DNA. This is double-stranded DNA. This is 5' and 3'. 3' and 5'. Because of the antiparallel nature. Now, DNA polymerase, DNA replication always takes place in the 5' to 3' direction. 5' to 3' direction. This is the direction of replication. That means DNA polymerase adds deoxyribonucleotides to the 3' end. Now, this DNA polymerase, it cannot initiate the DNA synthesis. A short stretch of RNA is formed here. This is a short stretch of RNA. This is RNA primer. This is RNA primer. This RNA primer is formed by enzyme RNA Primase. This is formed by enzyme RNA Primase. This short stretch of RNA is formed by RNA Primase. And to this RNA primer, DNA polymerase third can add deoxyribonucleotides. Then it would be like this. It would be 5' end. Then DNA polymerase will proceed like this. So it means that this is the free end of the newly synthesized DNA. At that site, RNA primer is formed. Why RNA primer is formed? This is RNA primer because DNA polymerase cannot initiate the DNA synthesis by itself because of the fact that it adds deoxyribonucleotides only to the 3' end. There should be a 3' end to which this DNA polymerase will add deoxyribonucleotides. In order to overcome this problem, in order to solve this problem, RNA primers are formed. Then this DNA polymerase will add deoxyribonucleotides to this RNA primer. So these are deoxyribonucleotides added by the DNA polymerase third, and this DNA synthesis continues. Then in case of another strand which is synthesized, this is called as leading strand. Another strand is synthesized in short stretches, and this is RNA primer, this is RNA primer, this is RNA primer. So these are called as RNA primers. The DNA polymerase has added deoxyribonucleotides to RNA primer. This strand is called as lagging strand. So DNA replication always takes place in the 5' direction. It would be 5' to 3', 5' to 3', 5' to 3'. So this is the direction of replication. This represents the direction of replication. Fork. This represents the direction of replication fork. This represents the direction of replication fork. That means replication will go in this direction, and in this direction, that is called as replication fork. And one strand is synthesized in the replication fork direction. This is leading strand, which is synthesized in the replication fork. Another strand is synthesized in a direction opposite to the direction of the replication fork. That is called as lagging strand. So one strand is synthesized in the direction of replication fork. Another strand is synthesized opposite to the direction of the replication fork. That is called as lagging strand. So these RNA primers, these are formed at two sites, at the replication fork site and at the free end of the new DNA strand. Then there is an enzyme that is called as Primase. Enzyme Primase. Primase is a complex of. It is a complex of this. Primase. It is a complex of RNA primer, RNA primase enzyme. It is a complex of enzymes, RNA primase enzyme plus helicase enzyme. We call this complex of enzymes which contains primase enzyme, RNA primase enzyme and helicase enzyme. Together it is called as primose. So it is a complex of enzymes, primose, and this primose is involved in the formation of RNA primer basically. And one of the important component of primose is this RNA primase, which is responsible for the synthesis of RNA primer. Then this priming phenomenon, it is a universal phenomenon. It is called as priming. Formation of RNA primer is called as priming. Priming is a universal phenomenon. Priming is universal phenomenon during DNA replication. That means during DNA replication it becomes indispensable. The formation of RNA primer becomes indispensable because DNA polymerase third, which is the most active enzyme in case of prokaryotes, it cannot initiate the DNA synthesis. It can add deoxyribonucleotides only to the 3' end. Now this primer, it gives a platform to the DNA polymerase third, and this DNA polymerase then adds deoxyribonucleotides to the 3' end of the RNA primer, and in this way DNA strand DNA synthesis proceeds ahead. DNA strand elongates. So we can say that this was regarding the then elongation of new DNA strand. Then we have third one is elongation of new DNA strand. That is, priming is a universal phenomenon. It is found in DNA replication of prokaryotes, occurs in viruses, where there is the formation of RNA primer, and to this RNA primer deoxyribonucleotides are being added by DNA polymerase third. the main enzyme for DNA synthesis. Then we have got elongation of DNA strand. Elongation of new DNA strand. Elongation of new DNA strand. This main enzyme DNA polymerase, it is third. It is the main enzyme. It is the main enzyme, main replication enzyme. Main replication enzyme in prokaryotes. And DNA polymerase alpha. DNA polymerase alpha, it is the main replication enzyme in eukaryotes. Main replication enzyme in eukaryotes, DNA polymerase alpha. These DNA polymerase alpha or DNA polymerase third, they add deoxyribonucleotides to this primer, and in this way this DNA strand elongates due to the antiparallel nature. That is, one strand is in the 5' to 3' direction, another is 3' to 5' direction. Because of the antiparallel nature of DNA, one strand is synthesized continuously and another strand is synthesized in short segments, what are known as Okazaki fragments. We have got two strands. One is called as leading strand and this is called as lagging strand. This leading strand, it is synthesized continuously in the 5' to 3' direction. 5' to 3' direction continuously. It is called as leading strand. And this leading strand is in the direction of replication fork. It runs in direction of replication fork. And the another strand, it is synthesized in short segments. Also, it is synthesis also takes place in the 5' to 3' direction, but it is synthesized discontinuously. It is synthesized discontinuously and in short segments, what are called as Okazaki fragments. And each Okazaki fragment is having the RNA primer. So it would be like this. We have this is one strand. This is, for example, 5' end. This is 3' end. This is 3' end, 5' end, 5', 3', 3', 5'. So this is parent DNA molecule, parent DNA strands. This is parent DNA strand. One strand of the DNA synthesized continuously in the 5' to 3' direction. We have 5' to 3'. This is called as leading strand. Called as leading strand. It is synthesized continuously. It is called as leading strand. And another strand is synthesized in short segments. This in short segments. These are called as Okazaki fragments. That means, obviously, this is 5' to 3', 5' to 3', 5' to 3', 5' to 3'. They're called as Okazaki fragments. They are synthesized in short. This is 5' to 3'. They are synthesized in short segments and discontinuously, what are known as Okazaki fragments. This is called as lagging strand. This strand is called as lagging strand. So this is the direction of replication fork. This is direction of replication fork. This is direction of replication fork. direction of replication fork. This is direction of replication fork. The direction of replication fork, that means one strand is synthesized continuously in the direction of replication fork. That means it runs in the direction of replication fork. The another strand, it runs opposite to the direction of the replication. This is the replication fork. The other strand runs opposite to the direction of the replication fork. That is called as leading strand. Obviously, here we have what type of proteins? These are called as single strand binding proteins. Single strand binding proteins. They prevent re-annealing of this single stranded DNA into double strand DNA. So these are called as single strand binding proteins. In addition to this, we have what type of enzyme? The main enzyme, DNA polymerase. This is DNA polymerase. DNA polymerase. DNA polymerase, also known as DNA replicases. DNA polymerase, also known as replicases. Then we have another enzyme that is called as primase. This is primase. This enzyme is essential for the formation of RNA primer. Where RNA primer would be? That would be here. These are RNA primers. This is RNA primer. These are RNA primers. Here also RNA primer. This is RNA primer. This is RNA primer. RNA primer. RNA primer. That means in each Okazaki fragment, RNA primer is formed. RNA primer is formed. RNA primer is formed. RNA primer is formed. Then we have this here we have enzymes what are called as helicases. These are helicases. They cause unwinding of the DNA duplex. In addition to this, we have Topo Isomerases. These are Topo Isomerases. They are Topo Isomerases. They relieve coiling tension ahead of the replication for these Topo Isomerases. Then this DNA polymerase, it continuously adds deoxyribonucleotides to the RNA primers in the leading strand as well as in the lagging strand. Then we have got these RNA primers. We have got RNA primers. These RNA primers are removed by DNA polymerase first. DNA polymerase first. These are removed by this RNA primer is removed, this RNA primer is removed, and this RNA primer is removed. And this DNA polymerase first also adds deoxyribonucleotides to these sites. Obviously, when you remove the RNA primer at the site, there would be a gap. This gap is being filled by DNA polymerase first, and then these deoxyribonucleotides are joined by enzyme DNA ligase by means of phosphodiester bond. This phosphodiester bond, it makes the DNA strand continuous, because after the removal of RNA primers, there will be gaps. These gaps are filled by DNA polymerase first with the help of deoxyribonucleotides, and this strand also becomes continuous and RNA primers are removed. This is what is called as how the elongation of DNA strand takes place. This is leading strand, lagging strand. Then we have termination of replication. Termination of replication. Termination of replication. Termination of replication. DNA replication terminates at a specific site that is known as terminus site. Terminus site. This terminus site, it represents where DNA replication stops. So this terminus site, it is having a specific sequence. It is having a specific sequence. This is having a specific sequence. What is called as TER sequence. This TER sequence, it serves as a substrate for this protein, TUS protein. TUS protein. That is, this TUS means terminus utilization substrate. And when the replication fork, it encounters this TUS complex, then replication halts. It would be like this. For example, we have this This is replication. This is replication direction of the replication fork replication. For example, here we have this terse sequence. This terse sequence, to this terse sequence which is on a DNA, TUS protein. For example, this is a TUS protein. TUS protein has attached here. This is TUS protein, that is terminus utilization substrate. This TUS protein binds to TUR sequence. When this replication encounters this complex TUR complex, this is called as TUR complex, the replication fork halts. This halts replication fork. Replication fork halts. That means replication stops. That means it acts as a, you can see that it acts as a hindrance for the replication fork to go ahead. It blocks the path of the replication fork. So we can say that it has resulted in the termination of the replication. Then we have got another, that is called as proofreading. We have proofreading. Proofreading. Although this DNA replication is accurate, it is accurate, but sometimes mismatched nucleotides occur erroneously. In order to remove these mismatched nucleotides, these DNA polymerase, all the DNA polymerases have got DNA polymerase first, second and third. They have got inbuilt, in case of prokaryotes, they have got inbuilt 3-5- exonuclease activity. Exonuclease activity. That means they can go, generally RNA polymerase moves in the 5' to 3' direction, but they can go, this is for example, they move in the 5' to 3' direction. This is 5' to 3' direction. They can go ahead, they can go behind, they can go in the reverse direction. They can go in the reverse direction, and that is in the 3' to 5' direction. And then they can pinpoint where the mismatched nucleotide is. Then they remove it, and correct deoxyribonucleotide is added there. In this way, it increases the fidelity of DNA replication. It increases the fidelity of DNA replication. What do you mean by fidelity of DNA replication? DNA replication is almost accurate, but sometimes mismatched nucleotides may get inserted. In order to overcome this problem, solve this problem, DNA polymerases can move in the 3' to 5' direction and can remove these mismatched nucleotides and put their correct nucleotides, so that DNA replication is 100% correct. Because if there will be one base or one nucleotide would be wrongly inserted, there would be a mutation that will change the the protein, amino acid composition of the protein. Ultimately that can change the biological character of the organism. So in order to have the correct DNA replication, these things ascertain that there should be no mismatched base pair in the DNA molecule. This increases the fidelity of the DNA replication. So in case of prokaryotes, this was about the DNA replication. In case of prokaryotes, we have 3 types of DNA polymerases. DNA polymerases are main enzymes of DNA replication. DNA polymerases. DNA polymerases are also known as DNA replicases. There are 3 types in case of prokaryotes: DNA polymerase first, DNA polymerase second, DNA polymerase second, DNA polymerase third. This DNA polymerase first, it's called as Korenberg enzyme. Korenberg enzyme. It was asked in examination. Korenberg enzyme. This DNA polymerase second, it repairs DNA damage. It repairs DNA damage. And third one, DNA polymerase third, it is the main replication enzyme in case of prokaryotes. It is the main enzyme of replication, main enzyme of DNA replication in case of prokaryotes. In case of eukaryotes, we have got, in case of eukaryotes, In case of eukaryotes, we have got DNA polymerases, what are called as DNA polymerases, and these are DNA polymerases are also called as DNA replicases, and there are 5 types of DNA polymerase. In case of eukaryotes, we have alpha, we have beta, we have gamma, we have delta, we have epsilon. We have DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase epsilon in case of eukaryotes. And among these, DNA polymerase alpha, it is the main enzyme of DNA replication in case of eukaryotes. It has got high polymerization speed, this DNA polymerase alpha. Same is the case, this DNA polymerase third, it has got the main replication enzyme, it has got high polymerization speed. So it is the main replication enzyme in case of prokaryotes, DNA polymerase third, and in eukaryotes, DNA polymerase alpha. And most of the replications, now this was regarding the most of the replication, that is bidirectional replication. Most replication are bidirectional replication. Most replication takes place in bidirectional. Bidirectional replication is the most replication type. Now what do you mean by this bidirectional replication? That means in bidirectional replication, 2 replication forks are formed. 2 replication forks formed, and these 2 replication forks, they move in opposite direction. And in case of it would be like this. In case of bidirectional replication would be this, like this. We have bidirectional replication is that type of replication in which 2 replication forks are formed and they would move in opposite direction. While as unidirectional replication is that replication in which only 1 replication is formed. In case of unidirectional replication, only 1 replication fork is formed. This most common type is bidirectional replication. Now we'll show the how this bidirectional replication takes place. This is, for example, suppose this is 5' end. Obviously this is 3' end, this is 3' end, this is 5' end, and this is replication, origin of replication. This is origin of replication. Obviously replication in the 2 directions. So this is 5' end. Obviously here what would be 3' end. Here 5' end. Here 3' end is present here. Here would be 5' end. And opposite to the 5' there is 3' end. Same is the case here. We have 3' end. Obviously here would be 5' end. And this is origin of replication, and this is 3' end. We have got 5' end here. There would be 3' end. Obviously here would be what? 5' end. So in case of this, we will take example of this. This is origin of replication. Origin of replication. One strand is synthesized continuously in the direction of replication. In the direction of replication. This is the direction of replication. This is origin. This is direction of replication. That means it is synthesized in 5' direction. That is, it would be like this. This origin of direction of replication. Direction of replication fork. Replication fork. So this, the another strand is synthesized in 5' to 3 direction, but that is in fragments. This is called as leading strand. This would be leading strand, and this is lagging strand. Lagging strand. 5' to 3', 5' to 3', 5' to 3'. This is synthesized in the direction of replication fork, and this is opposite to the direction of replication fork. Then is another. This you consider that it's an origin of replication. One strand is synthesized in the 5' to 3 direction. This is 5' to 3 direction. This is the direction of replication. Direction of replication. So origin, direction of replication, it is like this. That means 5' to 3 direction. It is a leading strand. Another is lagging strand. another is lagging strand. This is 5' to 3' direction, 5' to 3' 5' to 3' direction. So one strand is synthesized continuously, that is called leading strand. Another strand is synthesized in short segments, that is called as lagging strand. This is two replication forks. This is one replication fork, this is another replication fork. That means it is bidirectional replication, which is the most common type. So how many this is the origin of replication. So direction of replication fork is like this, so leading strand. And then we have got another, this one. You consider that this is origin of replication, direction of replication fork is in this direction. So in this direction this strand moves, so it becomes the leading strand. This is opposite to the direction of replication fork, this is called as lagging strand. This was regarding the mechanism of DNA replication. Thanks.